more about the process of post-transcriptional gene silencing, and now I'm going to call it as RNA silencing. So how does this process work? Double-strand RNAs within the cell are recognized by a class of enzymes called dicer-like enzymes. These enzymes cleave the double-strand RNAs to produce smaller RNAs that are 21 to 24 nucleotide long. One strand of this small RNAs then gets loaded into another class of enzymes called Argonaute. The Argonauts, in association with this small RNAs, are called RNA-induced silencing complexes, or RISC. The RISC then targets the mRNA by base pairing. At this step, there can be two effects. First, the RISC can cleave the RNA, that is called, and degrade the RNA, that is called RNA slicing. The other process that can take place is the RISC can block the translation of the RNA, and that is called translation repression. In both cases, the protein synthesis is blocked. And now this is again a simplified look of the RNA silencing mechanism that takes place within plants. Now, this particular RNA silencing process also can act as an antiviral defense response in plants. Now, in this case, the double-stranded RNA precursors that I just talked about in the previous slide are the viral or replication intermediates or secondary structures on viral RNA. So these structures that are formed during the multiplication cycle of the virus or the structures that are present on the RNA are recognized by the RNA silencing pathway, and then they are processed. Now, the role of RNA slicing as an antiviral defense response is well established. It has been observed with many, many viruses. However, the role of RNA silencing, the role of translation repression is not yet known. Now, when I joined my PhD lab, this was the concept. And luckily, while studying symptom recovery, what we discovered or what we found is that translation repression can also act as an antiviral weapon. So basically, this was the first time that we showed that translation repression acting as an antiviral defense response. And that is what I'm going to discuss in the next few slides. So the virus that I worked on was tomato ring spot virus. The tomato ring spot virus is an economically important pathogen. It has a wide host range, and it is economically important because it causes disease on economically important plants in Canada, like raspberries, peaches, etc. Tomato ring spot viruses are positive sense single-stranded RNA virus. And the viral genome is bipartite. Now, when I say bipartite, what I mean is that the viral genome is divided into two RNAs, RNA1 and RNA2. The RNA2 encodes structural proteins like coat protein, or here referred to as the CP. Now, tomato ring spot virus infected Nicotiana benthamiana plants also show this phenomenon of symptom recovery. What you observe is that in the inoculated plants, you do see this kind of symptoms. On the upper uninoculated leaves, after a few days of infection, you get very strong symptom development. And finally, new leaves emerge and they are symptomless. Now, symptom recovery, as I was discussing about the tobacco black ring virus and tobacco black ring virus and Nicotiana clevelandii interaction, where also we see symptom recovery. The reason was that you get viral RNA clearance. Now, in my PhD lab, this was before I joined my PhD lab, it was a postdoc called Juan Hogel, who tried to characterize symptom recovery in TORS-V infected plant. So his aim was that to basically see how recovery is taking place in tomato ring spot virus infected plants. And basically he looked at the viral RNA levels and RNA silencing and the RNA silencing mechanism. And he tried to see that whether the similar mechanisms are taking place as observed with the tobacco black ring virus. So the first thing he looked at was viral RNA. So this is a northern blot where you can look at the viral RNA levels. To his surprise, this was really surprising. What he found is that in the recovered leaves, there was no significant reduction of viral RNA. It was not what was shown with tobacco black ring virus. Then he looked at viral derived siRNAs, which are the hallmarks of RNA silencing. So by looking at viral derived siRNAs, you will be able to know that there's active RNA silencing taking place against the viral RNAs. And he did detect viral derived siRNAs, indicating that there is the active RNA silencing mechanism going on. And they published this work, but still the question remained is that in tomato ring spot virus infected plants, you see RNA silencing, but there's no viral RNA clearance. So what is causing recovery? So this was the question that I got, and it was a very interesting question. And so I took up this question and I tried to move forward with it. And to address this question, I used a tomato ring spot virus isolate called RASP1. Now, let me tell you why I took this isolate. Because when plants infected with this isolate when plants were infected with this isolate, they recovered, they showed the symptom recovery phenomenon only at 27 degrees centigrade. So at higher temperatures, plants were recovering. But at lower temperatures, plants succumb to infection and you would get, the plant will be dead. So this is one of the reasons why I selected this, because now I have a situation where I can basically compare between recovered plants and non-recovered plants. The other reason was that it was already known that at higher temperatures, you do get stronger RNA silencing effects than at lower temperatures. So the first experiment I did was I compared the viral RNA and the viral coat protein levels in plants infected by the virus, but kept at these two temperatures. Now so let me discuss the results that I got. First, let me discuss what I got at 21 degrees centigrade. So you see that at 21 degrees centigrade, we didn't get any recovery. We saw severe necrotic symptoms. When we looked at the viral RNA levels, the viral RNA kind of was present in all the samples that we tasted, and it showed a steady increase. A similar pattern was also observed with the coat proteins. Now we detected the coat proteins by using Western blots, and you can see here that there was a steady increase. So severe symptoms at 21 degrees centigrade were associated with steadily increasing levels of viral products. Now let's look at 27 degrees centigrade. What do you see at 27 degrees centigrade? You see a classical symptom recovery phenotype as observed by Juan. As observed by Juan's. So you do get recovery and then the recovered leaves have no symptoms. We first looked at the viral RNA levels and you do see viral RNA accumulation in the systemically infected leaves. And then you look at the recovered leaves. There also you do see viral RNA levels. And it was very similar to that we observed in the systemic leaves. So basically we are not getting any reduction in the viral RNA levels. Then we looked at the viral coat protein levels. And this was surprising because in the systemically infected leaves, you do see accumulation of coat proteins. But then in the recovered leaves, there's a drastic reduction of coat protein accumulation. However, the viral RNA levels were similar in the systemic and the recovered leaves. We also tested for the, we also examined the viral derived siRNAs in these plants, just to check whether there is active RNA silencing mechanism acting towards the virus. At 27 degrees centigrade at a higher temperature, at 27 degrees, viral derived siRNAs accumulated earlier and at much stronger levels than at 21 degrees centigrade. So to summarize, what we found is symptoms are associated with viral coat protein accumulation. Recovery is associated with reduction of viral coat protein levels at 27 degrees. Third, recovery is recovery is associated with stronger and earlier accumulation of viral derived siRNAs at a higher temperature. So based on this observation, what I did was, or not, I made a working model and asked two questions. This is just a part of my PhD research work. I will not go into further into the other details that I have done. So this was the first question that we asked. Is reduction of coat protein during recovery associated with translation repression? The second question was, is RNA silencing involved in the reduction of the coat protein and recovery of TORSV infected plants? So let us focus first on our first question. To do this, we thought we will look into the translational status of viral RNA during various stages of infection. And we thought we will compare the rate of coat protein synthesis in this different viral infection stages. Now, it sounded really straightforward, but then we had to think about ways to how we can do this experiment. So what we ended up doing is we ended up doing some in vivo labeling experiment. Now, what we did was we detached the leaves. That were at different stages of virus infection, and we fed them with radioactively labeled methionine for 2 hours. So during these 2 hours, suppose your viral RNA is actively getting translated. So the methionine will get incorporated into this newly synthesized coat proteins. However, if your viral RNA is not actively translated, then what will happen is you will get lesser number of coat proteins with radioactively labeled methionine. But this sounded straightforward, but here we have another problem, because during this 2 hours, you have other RNAs that are also getting translated, and they are also getting radio labeled. So we took another approach. What we did was we used immunoprecipitation. We used coat protein specific antibodies to separate out coat proteins from the unbound other cellular proteins. And then we ran them on a SDS page gel. Now, running them on a SDS page gel gives you some advantage. First of all, when you run them on an SDS page, and then suppose if you stain them with a Kumasi blue stain, What you can get is that will tell you the total amount of coat protein that is present within those leaf tissues that you analyze. So here you can see that there was a drastic reduction in the in the recovered leaves in comparison to the systemically infected leaves. So these two, so this basically corresponds to what we saw in our Western blots in a couple of slides before. Now, if you take this particular gel and then you expose it to a film, on the film, you will be able to detect the labeled coat proteins as and they will denote the newly synthesized coat proteins. And this will tell you about the translational status. So here you can see that there was strong labeling that took place in the early stages of infection and not at the later stages of infection. So we were observing translation repression of the viral RNA during late stages of infection. So this obviously was interesting to observe. Then we asked ourselves one more question: is that at 27 degrees centigrade we see this drastic reduction, but at 21 degrees centigrade we don't see the reduction; it's a steady increase. So we asked ourselves that how does the translational status in these particular systemically infected leaves look? So we did the same in vivo labeling experiment in systemically infected plants, in systemically infected leaves. So here you can see that these are systemically infected 6-DPI leaves at different temperatures. So at 21 degrees, and then the other one is at a higher temperature. At 21 degrees centigrade, you do see, when we looked at the total coat protein levels, it was pretty much similar at 21 and 27. When we looked at the radio labeled, when we looked at our autoradiograph film, there was active translation going on at 21 degrees centigrade, but at 27 degrees centigrade, there was repression. So translation repression of viral RNA2 was observed only at 27 degrees centigrade, but not at 21 degrees. So we got back to our working model, and we now have evidence showing that translation repression is taking place, and that is causing the reduction in the viral proteins. This was obviously interesting to find. Then we address our second question, that is RNA silencing involved in the reduction of the coat protein? Now, to address this question, we thought of an experiment where we would basically infect plants that are compromised in the RNA silencing pathway. And then we would look at whether these plants are able to recover. And also we would examine the coat protein levels in these plants. As you can see that the argonaute is one of the main components of the RISC complex, and that's what is required. That is what is required for RNA slicing or translation repression. So we selected argonaute as our target. Now in plants, as you all know, these are huge families of genes. We selected 3 different argonautes: Argonaute 1, Argonaute 2, and Argonaute 4. There was a reason for selecting these 3, because Argonaute 1 was shown to be involved in miRNA and siRNA mediated translation repression of host genes and slicing also. Argonaute 2 was involved in the antiviral defense response. And Argonaute 4, that was involved in the translation repression of another virus, potato virus X, but that was via resistance gene mediated pathway. So we basically selected plants that were lacking each of this gene, or not lacking, I would say, that were downregulated for these genes. Because we were doing this experiments in Benthammiana plants, and we had to use this particular approach called virus-induced gene silencing approach to silence this particular genes. So we inoculated the plants with the virus, and then we monitored them for recovery. Interestingly, Argonaut 1 silence plants did not recover from infection. When we looked at the viral RNA levels from these virus infected plants or TORS-V infected plants, we saw similar levels of viral RNA accumulation. But when we looked at the coat protein levels, it was interesting that Argonaut 1 silence plants only showed the accumulation of coat protein, while none of the other plants that actually recovered from infection uh, um, had coat protein accumulation in them. So this obviously showed that recovery and the reduction of the coat protein are Argonaut 1 dependent. Next, we asked ourselves that, is it because of translation repression? So we subjected this particular particular leaves to our in vivo labeling experiment. What was interesting was that the labeling, there was stronger labeling in the Argonaut 1 silence plants in comparison to the control plants, indicating that translation repression of viral RNA2 is dependent on Argonaut 1. So again, when we went back to our working model and now we have evidence showing that recovery is associated with Argonaut 1 dependent translation repression. And now we proposed a model where we think what is happening is in tomato ring spot virus infected plants, there is Argonaut 1 dependent or RNA silencing is somehow playing a role, and it is causing translation repression of the viral RNA2. And this translation repression is actually causing the reduction in the viral protein concentration. And once you have low levels of viral protein concentration, you are getting recovery. And this was the first example that we were able to show that translation repression of the viral genome can act as an antiviral defense response system in plants. So now we have discovered another weapon, a weapon in the plant that is being continuously used by the plants. So to summarize all of this data, what we observed is that the phenomenon of symptom recovery is influenced by temperature. It is also dependent on the concentration of the viral proteins. And finally, RNA silencing is involved in the symptom recovery phenomenon, that is for sure. And we did get translation repression by RNA silencing, and we showed that this is an antiviral defense response system. So this is a short story from my PhD work. If you want to get more idea about what else we did, we have published this work, and you can obviously read our paper. Now let's get into our final and last topic, that is, how can we apply the knowledge of plant-virus interaction? Now, obviously, we don't just generate knowledge for the sake of generating knowledge. There must be some use for it. And I think the knowledge that is generated from the plant-virus interaction in studies can be used in mainly 3 different ways. First of all, you can develop or design novel disease control strategies. I think now RNAi is being highly used to control viruses. Also, in plant-virus interaction studies, if we incorporate one more component, that is the vector, because that is what is causing the spread of the disease, right? So if we incorporate that particular component, now we have a way, or we can get some knowledge that will help us to find out ways to restrict the spread of the vector. And that will again help us to design novel disease control strategies. The second thing that we can do is we can take this knowledge and now we can basically make new models that will help us to predict outbreak of diseases. I think this is done for human diseases as well, but now we can obviously do it for the plant virus interaction studies. And finally, plant viruses are used as plant viral vectors in biotechnology or in biotechnology a lot. Now, when you are developing a plant viral vector, you just don't use it, or you don't use it based on the information that you have on the plant viruses. You also use the information on the plant part, right? So let me give you some examples about this, about the plant viral vectors. So as we all know, there is a burst or explosion to develop or to use the CRISPR-based systems. In line with the development of the CRISPR-based system, there is a great thirst to use plant viral vectors to deliver this CRISPR-based reagents. This is an active research. Here I am citing just 2 papers, but in the last 4 or 5 months, I saw like 5 or 6 papers that got published using different viruses to deliver CRISPR-based to deliver CRISPR reagents like Cas9 or the guide RNAs. So this is actively going on. Also, why I wanted to bring this topic is, so you found that I did my PhD on plant virus interaction. I'm still doing a little bit of plant virus interaction studies in addition to plant epigenetics. But what I would like to emphasize is that how I'm using my plant virus interaction expertise in my current lab. So in my current lab, what we are doing is we are developing this CRISPR-based or zinc finger-based targeted epigenome editing tools. We already have published a couple of papers, and I have been a part of these groups where we have developed tools like tools that will target DNA demethylation, that will basically cause targeted DNA demethylation, or we have other tools for targeted DNA methylation. There are more tools that are in the pipeline, and they will soon get published, or in the next 6-7 months, they will get published. So we are developing this tool, and I'm lucky to be a part of this, this group, where I am, on one hand, I am developing these tools, but on the other hand, we also aim to use plant viruses to deliver these reagents for epigenome editing. So that is something I'm actively doing in my current lab. And at this point, I would say that my plant virus interaction knowledge that I had from my PhD is helping me a lot. So you saw that how plant virus interaction can be used. Now, moving on to my my next couple of final slides. So this is